Our first talk is on the topic of personalized neuroscience for developing mental health applications. Our speaker is Dr. Leanne Williams. Dr. Williams is the founding director of the Stanford Center for Precision Mental Health and Wellness and of the Stanford Pan Lab for Precision Psychiatry and Translational Neuroscience. She is the associate chair of translational neuroscience in the Department of Psychiatry and Behavioral Sciences and director of education and precision medicine at the Palo Alto VA Mental Illness Research, Education, and Clinical Center. Prior to joining the Stanford community, Dr. Williams was the founding chair of cognitive neuropsychiatry and directed the Brain Dynamics Center at Sydney Medical School. Her PhD was completed with a British Council scholarship for study at Oxford University. Dr. Williams' Center and translational programs integrate advanced neuroimaging technology and digital innovation to transform the way we detect mental disorders, tailor interventions, and promote wellness. She has developed the first taxonomy for depression and anxiety that quantifies brain circuits for diagnostic precision and prediction. Dr. Williams' research programs are supported by funding from the National Institutes of Health, spanning initiatives including priority research domain criteria, human connectome, and science of behavior change. She has contributed over 325 scientific papers to the field. Professor Williams, please, we could switch over your slides now and look forward to your presentation.
Thank you so much, Angela, and for the opportunity to be part of this wonderful eWear Symposium and to share what we're excited about in translational neuroscience for wearables. Let me just do a sound check and check that the sound is coming through.
I can hear you great.
Okay, so what I'm talking about today is very much about where the field is accelerating in exciting ways for understanding mental health from an objective measurement-based way, particularly in regard to how the brain functions. And what I want to suggest to you is this is important for when you're thinking about developing wearables, particularly in this space of mental health and wellness. First of all, to acknowledge that the work that I'm presenting was a really exciting and stimulating and productive collaboration with Shannon Bauf, who is, as you know, professor and chair of chemical engineering, and several of the colleagues listed here. And what I'm presenting will also be a foundation for Dr. Khan's presentation to come. So this was very much the kind of neuroscience basis of the project. I'm focusing in particular on depression and anxiety, and more broadly disorders of mood and anxiety that are result from, at least in one form, result from prolonged stress that is outside of our control. And I think most of us would know this, but it's just to remind ourselves just what an impact these conditions have, even though they still remain stigmatized and hidden. So we're talking well over 300 million people affected across the world and a rate of 1 million suicides every year. We were talking a little earlier between the panelists how the COVID pandemic is one of those prolonged, uncontrollable stresses that's unusually occurring for all of us that can lead to exacerbating these kinds of disorders and symptoms. What the current situation is one that we really want to change, and what I will propose today is how we can move from this current situation to using wearables in a way that takes account of some connecting pieces. So the current situation, as you may know, is that depression and anxiety are diagnosed and assessed through checklists. So these are diagnostic statistical manual where we have a list of symptoms that we ask the patient or the person about. And we're looking for something like, are there 5 of these present at once? So there's not a test, there's not a biological measure. There's not any way to monitor these objectively right now. What I want you to note is when you're looking at a diagnosis of depression or anxiety, there are distinct features, like in blue, depressive symptoms for depression, excessive anxiety and worry for anxiety, but many that are common. So concentration problems are one diagnostic feature that's common and one that's very prominent during the pandemic. Others I won't talk about today, like fatigue and restlessness, also common. What I'm leading and excited about is a new way to think about how we understand and diagnose these disorders and provide the foundation for wearables. And it is to move to what you could think of as precision medicine for mental health and psychiatry. So these are kind of icons to say, we have our current situation of checklists, but the organ of interest when we're thinking about mental health is the brain. These are essentially disorders of how the brain functions. And they are expressed in our behaviors and our feelings, emotions, and experiences. So what we're doing in my work is we're actually going to that direct proximal organ and asking what ways can it be disrupted that produce that checklist of symptoms. And in doing that, we can see these individualized patterns, like that are distinct ways that we can experience depression and anxiety, rather than thinking of them as all the same. And that gives us a foundation for interpreting the signals that we could then gather in a more distal way using wearables, and ultimately a way to individualize all of how we explain, like diagnostically, monitor, and ultimately treatment over on the right here. How I assess the brain in real time in action is using functional MRI, and that gives us a time series of what the brain is doing both at rest and in response to different tasks. So a little bit like a stress test that you might do in the cardiac setting. And what I've discovered is that, at least for now, what is consistent, likely there'll be more and different combinations, but there are at least 6 ways that the brain essentially can go awry and create different combinations of symptoms and experiences that meet the diagnostic criteria. So we could look at 3 of these in more detail, but to orient you, the colors here are referring to what are known as large-scale circuits of the brain. So the blobs are representing regions of the brain that are particularly relevant to how we think and feel. These are largely unique to humans. There's one, the purple one, that we can also see homologies in animals. But they are like ways that regions of the brain connect and correlate to support certain functions. So the blue one, for example, is a set of 5 regions that connect together when our brain is in a free association mode called the default mode. Whereas the red one over on the right is a largely different set of regions, not completely, but a different set of regions that are engaged when we're focusing our cognitive functions and more planning. And you'll see there's some common regions, and that they operate as hubs that help communicate between the circuits. So the different dashed lines and size of the dots are representing the nature of the dysfunction. Like either the activation is too much, and the dashed lines are where there's a breakdown in the communication. So if we go further, we can look at 3 that I'll talk about a little more in relation to wearables. So as I said, that blue one, the circuit involved is called the default mode. There's a lot of evidence, particularly over the last 5 years. There's a project called the Human Connectome Project that we are part of, and is like a human brain variation of the Human Genome Project, where we're mapping the whole of the human brain and its connections. And we find the default mode is really important to human function. It seems particular to humans. And in this free association state. What's important is it is anti-correlated with other circuits of the brain. So if you're engaged in this restful mode, that tends to be when you can reflect on your internal self. It means that your concentration circuit is actually not active. So they're in an anti-correlated relationship. Under situations of depression, this default mode becomes stuck. It stays overconnected. So there's too much self-reflection or too little, which makes a state of worry and rumination and focus on depressed negative thoughts. This orange one we call negative affect. It is particularly responsive to detecting potential threats in our environment. And in anxious form of depression, where you have a physical signs of anxiety, you'll have a breakdown of this circuit. You'll have too much response to threat, the bigger dots, and a breakdown in the connection with the part of the brain that regulates that. So we think of that as particularly about anxiety and acute stress. This one over here, 6, is called the cognitive control circuit. Some people call it the executive circuit because it's so involved in focused planning and thinking. The concentration problems I talked about that are common to depression and anxiety tend to be reflected in a breakdown of this circuit. And in some people, you'll get both biotype 1 and 6 occurring because the anti-correlation between them gets disrupted. In order to get a standardized way of quantifying these, this is a big challenge for the field because we don't currently have this for brain health, particularly mental health. We have a way to clinically read the scans, but not get numbers that can go into tests. So this has been a major effort in my lab and leads us, I believe, to a platform from which we can develop peripheral wearable measures. So what we've done, we've collected multiple large data sets where we've quantified these large-scale circuits and quantified the dysfunction in people experiencing depression, anxiety. And then we've developed a standardized way to define the regions and the strength of the connection between them and the extent of activation. And that allows us to then create literally numerical values. So we can extract from the image a numerical value. And in that sense, we get a value of what we call a biotype score of, let's say, this default mode relating to depressed mood, this second, this biotype here, the orange one relating to anxiety, and biotype 6, we can get a different score relating to concentration problems. And what was encouraging is we find those scores are independent of each other. So we're not getting overlapping information. It is distinct. Also to note, for the concentration problems, we see a different type of dysfunction that correlates as well and explains a different source of variance, which is this circuit involved in sustaining attention over time. So not surprisingly, if that's disconnected, indicated by the dashed lines, it also relates to concentration problems. What's inspired this approach in many ways is the huge progress that's been possible in other fields of medicine. Particularly, you can think of this timeline. So I've been looking for inspiration because obviously we're dealing with complexity when we're talking about the brain, complexity when we're talking about mental health, complexity in terms of measurement. So what can we learn from other complex stigmatized areas of health? So if I go back in time, I see 1948 was when we got this incredible change in how we understand cardiovascular health, heart health. Not that long ago, 72 years, a few more now. And that before that, heart disease was a second leading cause. Half of older adults died from heart disease in the US, and that is dramatically reduced. And that was a result of a huge effort that was to really understand the heart, the proximal organ of interest, and ultimately from there develop the suite of tools that we have now, imaging right through to apps and wearables. And I'll touch on that a bit more. There's other innovations that we take inspiration from. So breast cancer, we have precision medicine approaches now because of the discovery of the particular receptors and the Herceptin related subtypes. Also with diabetes, the discovery of ways to monitor insulin have made a big difference. And glucose, I should say, and insulin. And then with HIV, it was not treatable until we actually discovered the particular genetic variant and receptor we needed to target. So now I think with the Human Connectome Project and other advances, this is time to do the same. So cancer, I think, is one area where we can conceptualize it and visualize. We have types that we can now measure with imaging of the tumor itself, getting genetic information, but it's not quite as, it's complex and important, not quite as complex as an illness like cardiovascular disease, which has a lot of environmental factors and stress factors. So in some ways, I think about cardiovascular disease as a good kind of framework to think about mental health. And to go back to the Framingham study, it is kind of striking that nothing was known in 1948. So it was actually the death of FDR that initiated this huge study, what became a huge study in Framingham, Massachusetts, which was to say, okay, there's clearly a problem. Half of older adults are dying from heart disease. We don't know how to treat it. We don't understand it. So let's launch this study, and that became the National Heart, Lung and Blood Institute of the NIH. What they did was really important. So the history of it is that FDR died with a blood pressure of reading of 300 over 190, which is extraordinarily high. And the three measurements before that just got higher and higher. And they were giving him the wrong, as we know now, the wrong treatments, including digitalis. And so it prompted this, like, we need to understand more. And that study, the Framingham, has led to this understanding of the heart itself, imaging modalities, many sophisticated ones, that led to the capacity to develop sensors and wearables to peripherally assess the heart, ultimately to apps. Obviously don't have the same precision, but they can tell you when you likely need the imaging. And that's given a way to understand individual profiles and treatments and matching of treatments. So that's my vision for mental health. Like, let's understand the brain, let's image it, get detailed pictures. That will give us a way to think about wearables that link to those measures. Maybe we can then move that to apps, and we can have both distal and proximal measures to subtype and intervene in a more effective way. And the way we've thought about it in this project is linking from symptoms to the biotypes to sensors to get to that. And here I give a shout out to Dr. Khan, who's done an amazing job of actually developing the sensor, which he will talk about. But to get there, we've had to connect this right chain neuroimaging biotypes with the physiology as it would normally be measured in the lab as the foundation. So we looked at, like, what do we know from current knowledge about what physiological measures do relate to these brain circuits. And this is the kind of list we came up with and the crosslinks. I'm going to give you an example of where we've got to with heart rate variability. And there's a reason why that particular measure is important. Cortisol is incredibly important because it's the stress hormone related to the pathways to depression. We're collecting those data now, but we didn't have it available in time to analyze for today. The heart rate variability information we've included in this recent publication and review, which will be available for open access soon. And what we did was, while we were recording in the MRI scanner the brain circuits, we also recorded heart rate, and we recorded it from this PPG signal. So it's recorded at the same time as we're scanning the brain. And we can extract several measures of heart rate variability. So the root mean square of successive differences of the beat, those interbeat intervals that are more than 50 milliseconds, so a different way of looking at variability, the standard deviation of them, low frequency, high frequency, and the ratio of them. It was important to break these down because in depression, there's good evidence actually that, sorry to go back one, there's good evidence that depression is characterized by low variability, and that's thought to be related to the disruption of the brain circuits that regulate it. But we don't really know which of these is more important, and we want to know for developing wearables. So how we went about this is we had measurements of the 3 biotypes I've been focusing on. We had measurements of those heart rate metrics, and then we did a tree-based classification model to look at what aspect of those heart rate metrics and what aspects of the circuits biotypes related to the symptoms. And this is it's a busy slide, so I will walk through it. It was encouraging that the initial model does improve over accuracy by chance accuracy by 29%, and that's in the validation set. So let me walk you through. So if we look at depressed mood, that was actually predicted most importantly by this default mode circuit biotype. So the colored blue dots on this figure are SHAPS values of the relative importance in the model of these different measures. There are other circuits involved, so we could go into why they are there and what are the interactions, but I'll highlight the ones that we predicted. You can then see that this ratio of low frequency, high frequency in the heart rate variability metrics also important. So those two are going together in predicting depressed mood. If we then look at anxiety, we see this negative affect orange circuit is really important. It's coming out up top. We see the same ratio, low frequency, high frequency, also important. So that seems generally important for distinguishing these symptoms. But for this type of symptom, low frequency is also really important. For the concentration problems, we see the cognitive control circuit biotype, which we expect, and interestingly also the attention one, so they're contributing importantly together. We see now the same ratio, low frequency, high frequency, but in this case, the high frequency component of heart rate variability is particularly important, unlike for anxiety. So this gives us an initial hint that we can get some precision over what measures we would want to include in a wearable. And one of my goals would be, how do we do this so we can do better for treatment outcomes? And as many of you may know, right now, it's pretty much trial and error because of the checklist system. So only about 1/3, 3 in 10 people actually remit on the first treatment, and usually it's a 2 to 3 year trial and error process. So we really want to at least double that by using these measures. And so far in separate work, which I won't go into, we are finding that knowing about the biotypes can get us to 75 to 81% accuracy. And I believe that adding in the wearables will only refine that and certainly give us granularity in real-time measurement. So to wrap up with envisioning the future, I would imagine we would have some kind of clinic of the future where we could bring all this together. We'd have the biotype information, the real-time wearable. We'd be able to track how people are doing, like in their daily life, and then know when we get a kind of ping, so to speak, when they may need to come in for another scan. And to Yasa Khan, who took so when we do the biotypes, the heart rate variability, we're able to include this incredible wearable as well. So let me stop there and thank you all again for the opportunity and for your attention, and I would really welcome any thoughts and feedback and questions.
Thank you so much, Professor Williams. This is exciting research and certainly so important to be able to characterize and quantify what's happening in the brain. So I want to encourage folks, please use the Q&A, type in your questions. We'll have time for just a couple of them right now. Professor Williams, you were showing a type of brain dysregulation around cognition and attention. Is that the same as ADHD? And do you believe that these interventions are going to be useful for ADHD?
That's such a good question. I'm glad you asked it, Angela. So that circuit I'm showing, particularly the attention, the yellow one, is also disrupted in ADHD. And although we need more evidence, there's an indication that ADHD in child, adolescent times in life, in some people is expressed as anxiety, depression in adulthood. So what it may be the case is there's a form of that kind of concentration disruption that expresses itself in children more in the symptoms of ADHD, but later on we see it as a concentration form of depression. And right now we've started a trial where we're using it as a pharmacological intervention for treating that type of depression in adults. It's the same pharmacological intervention that is used in children for ADHD. And in prior work, when I have looked at children with ADHD, those children that respond best to this intervention actually have the concentration problems and anxiety present. And it's another sign of a little bit of siloing in the field, whereas it would be unusual to assess for both ADHD and anxiety, depression at the same time. But I think there's value in doing that.
Definitely. Thank you.